Thanks a lot for the invitation, Helmut. Thanks a lot for giving me the chance to present our data here in a plenary lecture on this fantastic conference. So I will present mainly two parts of work. One goes back to my time in Stefan's lab in Göttingen, where we started all the lipid membrane work, and continuing it now in my new post in the Weatherall Institute of Molecular Medicine, where as a physics person, I'm in the middle of medical people with a lot of fantastic opportunities to start very good projects. Now, it's all about microscopy, optical microscopy, and one of the main driving forces here is to really be live cell compatible, least invasive, and also to be able to go inside the living cell. And for that we use far-field microscopy, or focusing of light. And when we do focusing of light, we send a laser into a microscope objective lens, which produces this small spot size. And here we excite the fluorescence and we detect the fluorescence onto a detector. Now the problem with far-field optics, or with using focused light, is that the size of the focus, or the point spread function, how we call it, is limited by the diffraction of light. And it's given by this famous equation from Ernst Abbe, back from 1870s, which is ruled by the size of the spot is ruled by the wavelengths of light used and by the focusing strengths of the microscope objective. So for visible light, and for the best lenses around, it's roughly about 200 nanometers along the lateral direction and 600 nanometers along the axial direction, the size of this focus. Now, what's the problem with having a focus that is that large? Well, if we scan an object with that, and we image it onto a detector, we cannot distinguish alike objects that are closer together than 200 nanometers, because we cannot separate them at once in time and space. So we end up with a blurred image. So what's the solution? Well, if we would have a smaller spot, we could distinguish these objects in space and time. Now, how can we do this? How can we distinguish objects that are closer together than these 200 nanometers? Well, we can't do much about this equation because it's the physical limit and we don't want to move into UV or something like this. But we work with fluorescence and we can modulate the fluorescence. We can certainly turn it on and off. So imagine we would have this 200 nanometer spots and we have all these objects inside there that we want to distinguish. Imagine we would find a mechanism where we switch off or inhibit the fluorescence everywhere but at a single spot. Then we would end up with an observation area and thereby a spatial resolution that is much smaller than these 200 nanometers. Now this is the principle of STAT, and what we have to do is really we have to find a mechanism and a way to inhibit the fluorescence in the outer part of our focal spot. Now let's start again with our far-field microscope, in this case a confocal microscope, for example. So we have our laser, we send it into the sample, we focus it to this 200 nanometer large spot. In there we excite all the fluorophores or the fluorescent labels to their excited state, and when they de-excite, they send out spontaneous fluorescence emission, which we detect on a detector, which gives us the signal. Now, if we add a red laser, I will call it STAT laser later on. So the red laser means in the red part of the emission spectrum of our fluorescent label. And it hits on the excited state. It stimulates the molecule down to the ground state, thereby emitting another stimulated photon, but we don't detect that. So the result of that is that the spontaneous fluorescence that we detect is inhibited because we have another process that is driving the molecules down to the ground state. So if we plot the spontaneous fluorescence that we detect over the intensity of the STAT laser, what we see is that we can nicely switch on and off the fluorescence. And more importantly, we can drive this process into saturation. Now, so far so good. We have a dark microscope because we switched everything off. But if we modify the STAT laser in such a way that when it's focused, so we insert an optical element, that when we focus it, we end up with a donut-like intensity distribution. We have exactly what we want, because now the STAT laser is only active in the outer part. But this one is also ruled by diffraction. So it's also further apart than 200 nanometers. But this process is driven into saturation. So if we crank up the power of the STAT laser, the area in which fluorescence is still allowed, which is the green area here, is getting smaller and smaller and smaller. And that's one basic feature of our the STAT microscope, because we can tune the diameter and thereby the spatial resolution of our microscope with the intensity of the STAT laser. Like shown here for fluorescent beads, if we increase the stat intensity, we see that we can reveal details of the sample much, much better if we increase the intensity of our stat laser, and we can continuously tune the size of the resolution or the observation spot size. It follows this square root law on the intensity. And this will be a key feature later on in my experiments. Because what we wanted to look, and we started that years ago, is to look on mesoscale plasma membrane organization, because it's a very important mechanism there, because the molecules in the plasma membrane trigger a lot of cellular events. Molecules are heterogeneously distributed. They interact with each other, lipids with proteins, proteins with proteins, and we have interaction with the underlying cytoskeleton. And everything is on very small spatial scales. And that's how we got attracted to this, to investigate it maybe with our stat microscope. So, especially if we talk about plasma membrane organization, you always come into one comment, okay, is this a lipid raft or not? So what are lipid rafts or lipid nanodomains? It came up, if we look into the literature, it's supposed to be transient cholesterol and sphingolipid enriched domains. They bring together molecules on very small spatial scales, so it's densely molecular packed. And by bringing these molecules together, they compartmentalize cellular processes. Now what's the problem here? Well, the problem is it's highly heterogeneous, it's highly dynamic, it's very small, smaller than 200 nanometers if you look into the literature. And you remember 200 nanometer was the diffraction limit. And that's why a lot of techniques miss the temporal and spatial resolution to directly investigate these events. And that's why it's still highly debated. So we started years ago trying to investigate the magic of these nanodomains. So we took live mammalian cells at room temperature, at physiological conditions, and incorporated fluorescent molecules into the outer leaflet of our cells. In this case, we labeled a lipid with an organic dye, for example, the organic dye ATO647N, but we're using other dyes as well. I will come to that later. And via BSA complex, we efficiently deliver them to the outer leaflet of our plasma membrane. So we started off with two lipids. One is a phospholipid, phosphoethanolamine. I will call it PE later on. And a sphingolipid, sphingomyelin. I will call it SM later on. Now remember the definition of these nanodomains, sphingolipid-enriched domains. So we were expecting, that's why we choose these, we were expecting maybe we see a difference between these two. So we incorporated these lipids into the plasma membrane, and then we imaged them with our confocal or stat microscope, and we saw a very homogeneous distribution here of the lipids. No domains or something like that. Well, it's live cell, so the lipids diffuse very, very fast. And if there are any transient domains or something like this, maybe we have limited temporal resolution. We can't resolve it with our microscope. So instead of imaging it, let's discover the diffusion dynamics. Let's use exactly what is hindering us to image it. Let's investigate the diffusion dynamics, because from diffusion, you can extract a lot of parameters on molecular interaction as well. So instead of scanning the spot over our plasma membrane, what we did now is we just parked our observation spot on a certain area or a certain point on the plasma membrane and just waited for fluorescent lipids to diffuse through there. This is, for example, the signal if we lower the concentration of the fluorescent lipids pretty much. Most of the time, there are no fluorescent lipid analogs in our observation spot. So this is fluorescence over time. You see most of the time noise. But from time to time, these fluorescent lipids diffuse through there, give rise to some signal, and when they move out, the signal is gone. So we have these fluorescent bursts. And what we now want to determine is how long did it take the molecules to diffuse through our observation spot, and thereby determine molecular mobilities. Now there's a very nice technique around with which you can do exactly this. It's fluorescence correlation spectroscopy. So what you do is you correlate this fluctuating time signal, and you end up with a decaying curve where the decay time represents the characteristic time for the process that is causing the fluctuations. In our case, it's diffusion. So the inflection point is basically the average transit time of the molecules through our observation spot. From that, we can determine molecular diffusion coefficients and thereby molecular mobilities. Now, remember, with our stat microscope, we have the unique possibility to tune our observation spot just with the intensity of our added stat laser. So our experiments look like this. We call it stat FCS experiments. What we do is we record FCS data. at different stat intensities, so for different sizes of our observation spot. And we determine the transit time for each of these measurements. Then we calculate the apparent diffusion coefficient and plot the dependency of this apparent diffusion coefficient on the diameter of our observation spot. And this dependency varies for different diffusion modes. Let me guide you through that. So this is just a sketch. Apparent diffusion coefficient, observation spot diameter. For free diffusion, we expect a constant. Because no matter at which spatial scale we look at the diffusion or we check the diffusion, it should be the same because it's free diffusion. If we have trapping, trapping is the molecule moves, interacts with something, stays there for a while because the interaction partner is slowly moving or immobilized. It stays there for a while, moves on, again is trapped, moves on. We would expect the red line because the smaller the observation spot, the more the transit is ruled by the trapping interaction, which apparently slows down the diffusion. Now, if we have this domain incorporation, this nanodomain idea, so the molecules move, get into this highly ordered molecular packed domain of certain size, it's trapped in there, but it still can move within there. So what we would expect then is also a very similar dependency as for the trapping, but we would expect a kink roughly at the size of the domain. Now let's see what we see for our lipids. This is the data, diffusion coefficient, observation spot diameter, PE, SM. And what you clearly see, for the PE, it's close to constant dependency. So we really have free diffusion, only very weak interactions, weak trapping. While for the sphingomyelin, we have this strong going down, really clearly going down. So we really have trapping. So the sphingomyelin really moves, interacts with something, moves on, interacts with something. Now we can take this data, analyze it a little bit more closer, and we can find out that this is a complex on the molecular scale, probably with slowly moving proteins and so on, because it takes roughly 10 milliseconds and there's no movement during trapping. It looks like it's really fixed at that point during these 10 milliseconds. It's cholesterol assisted, because if you treat the plasma membrane with cholesterol depleting reagents like cholesterol oxidase, we see that the trapping is released. We hardly see trapping anymore. Also, it seems that the binding partner is bound to the actin cytoskeleton, because if we treat the plasma membrane with drugs that deplete the level of the actin cytoskeleton, it depolarizes, just like, for example, latrunculin B, we also see that the trapping is less. But we don't see any sign of nanodomain incorporation. So to investigate that further, we first looked at a whole gamut of different lipids. So we gave our organic chemists a really hard time because we synthesized all these different fluids and lipid analogs carrying different dyes at different positions, sphingolipids, phospholipids, saturated, unsaturated, gangliosides, and so on. First of all, what we saw is, at least for the molecules investigated, we didn't see a dependence on the dye or label position. On the other hand, the interaction differs strongly between the different lipids. So the moment we change something on the structure, we see a difference in trapping strengths and dependency on cholesterol and actin. Especially the ceramide group and the polar head groups, the moment we had those, we had a trapping interaction. So clearly, what we see here is a specific lipid-protein interaction. And we now have to find out what is the binding partner. We're really going to dig into this. Now, another thing to find out more about these trapping interactions is, so far we only investigated, we averaged from FCS data taken at different time points and different spatial points. Now it would be great to be able to record FCS data simultaneously at different points to compare, is there spatial heterogeneity in these interactions. So we took the idea of scanning FCS and applied it to scanning stat FCS. So what you do is you either scan your beam very fast, 4 kilohertz or something like this, in a circle or a line. And thereby you can record FCS data for each pixel along this line, because you scan it so fast. So at the end, you end up with a diffusion map, where this is the space, so every pixel along the line or the circle, and this is mobility, which is the decaying FCS curve, from red to blue. And the yellowish area is roughly the inflection point, which is the average transit time. So if we have fluctuations in this position, we know there is some heterogeneity. So let's again compare PE versus SM. Confocal recordings for PE, some heterogeneities, but not very strong. For SM, a little bit more pronounced, but not really distinct. Now let's go to STET recordings, 80 nanometer large spot. For PE, again, we see very homogeneous distribution, which is in line with our previous observation of free diffusion. But for the sphingomyelin, we see that suddenly for the STET recording. We see these peaks showing up. And these peaks are smaller than 80 nanometers in size, so smaller than the resolution, and they are so slowed down diffusion, spatially distinct. And it's only visible in the STET recordings. Now, if we look back into our previous data, if we only have slowed down diffusion in the STET recordings, it's clearly trapping. So these are the trapping sites. This is where the molecules interact with other molecules, stay there for a while, and then move on. Now, in the next step, we wanted to see, are these stable, these trapping sites, or do they move in space? So we recorded subsequent 10-second measurements of these mobility maps over time. So this was the first one. We had these trapping sites. And if we look on the next 10 seconds and the next 10 seconds, you see that the number of trapping sites as well as their position changes. So they are transient or mobile or so. And they are transient in the second time range because they have to rule. To be able to rule a 10-second measurement, they have to persist at least for a few seconds. Now, the current pictures, and I took this graph from one of Kai Simon's reviews, is that a protein is not alone. It's interacting with lipids and cholesterol. And especially, it seems that we have formed transient sites, which may be this, where these are smaller than 80 nanometers, few seconds lifetime. We have a constant exchange of lipids. Lipids move in there. Only certain lipids stay there for 10 milliseconds, move on. It's actin dependent, it's cholesterol assisted, and it's sphingolipid specific. So you could say, wait, we heard this before. If we go back into our definition here, cholesterol and sphingolipid enriched domains, could be. Now, densely molecular packed, highly ordered, a lot of molecules together. Well, let's investigate that further. There's a biophysical model for this, because domains of higher order. There's a putative physical model system for these lipid rafts. So you just take a model membrane, a single bilayer, a normal bilayer, you just put it on a support, and you make a mixture of lipids in there. For example, if you do a ternary mixture, they can phase separate. They separate in a fluid environment and a more ordered environment. And this more disordered environment or fluid environment is usually filled up with unsaturated lipids. Unsaturated lipids have a kink in their chain, so they need space. That's why it's more fluid. And then there's the more ordered environment. I will call it LO and LD later on. So in the LO environment, we have saturated lipids, which don't require that much space because they don't have a kink in their chains. And this LO environment is often taken as a raft model, because it's highly molecular ordered. So let's see, let's compare this characteristic with our live cell nanotrapping or STET FCS recordings. So let's take PE versus SM again. The STET FCS data in the living cell shows vast difference between these two lipids. If we look where these lipids go into these phase-separated model membranes, we see that both of them mainly go into the LD environment, so the non-raft environment. 95% go in there. Also, the labeled SM, it strongly interacts in the living cell, but it hardly goes into the raft environment in the model membranes. So clearly, we can say it's a different nature of the nanoscale trapping that we investigated before for STET FCS and the preferences for certain environments, especially the LO environment in model membranes. But we have a problem here. All of our fluorescent lipid analogs hardly go into the LO environment. They only go there 5%. We hardly, if there's an LO environment in the living cell, we wouldn't see it, because our lipids don't go in there. So, starting with these lipids, which mainly go into the LD environment, we started off investigating other lipids. How do they partition? What preference do they have? As I said, changing label positions, like putting the dye on the head group, didn't make much difference. We also saw that this was mainly going into the LD environment. Now, of course, the dye, it's large and it's interacting, it needs space, so that's why it doesn't go into the LD environment, LO environment. That's why we thought, let's put a linker in between, a linker between the lipid and the dye. So we put in, starting off with this head group labeled molecule, we put a PEG linker in between. So we put the dye away from the membrane. And surprise, surprise, 70% it goes into the LO environment. To have a comparison, we used an unsaturated lipid as well, of the same kind. It has a kink in here, and as expected, it only goes 10% into the LO environment. And just to have another comparison, we used a PEC-linked cholesterol, which goes 50-50 into the LO-LD environment. So we have 4 different lipids, phospholipids, that have all different preferences for different ordered environments. So if it has an effect on on our live recordings, we should see a difference. But this is what we saw: diffusion coefficient, observation spot diameter, straight line for all of them. So they all diffuse freely. While for the sphingomyelin, remember, really is going down. Also, we looked on the partitioning characteristic of some sphingomyelins. And this is the trapping time, basically, plotted for different fluorescent SM analogs. And what you see also there is no correlation between the trapping time and the preferences for the LO environment. So clearly we can say partitioning in model membranes has no significant effect on the investigated live cell dynamics and interaction, at least what we investigate with our StatFCS recordings. And from that we can say, okay, for the lipids that we've seen, there are no stable model membrane LO-like lipid domains in the undisturbed and investigated living cell. Now, to confuse you more, because you might now think no lipid rafts, but this is not true, because we now do a lot of work on these giant plasma membrane vesicles. And these are vesicles budding off a living cell. And you can then separate them. So you have really vesicles. And these vesicles are really nice nanoscale model systems because they are full of the cellular plasma membrane proteins and lipid diversity. And with this, we can decipher the influence of lipids and proteins on their functionality. Now, the funny thing is, if you treat them in a certain way or you cool them down, these GPMVs also phase separate. They phase separate into a more ordered and a more disordered environment. And proteins tend to segregate into one of the phases, either LD or LO, depending on the protein. So the lipid environment has a function. Just to show you this, we investigated also the GPI-anchored protein. This is a protein on top. The dye sits on the protein, you have a linker, and then you have a lipid anchor that anchors the molecule to the plasma membrane. And this one, in these GPMVs, goes mainly to the LO environment. And if we investigate that with our StatFCS recording, shown here, diffusion coefficient, observation spot diameter, again, PE, SM, and the GPI also shows trapping, but it shows this kink, roughly at 100 nanometers. So we have weak partitionings into roughly 100 nanometer large domains. So our message from all of these measurements is, don't call everything lipid rafts. Call it what it really is. Lipid-protein interactions like we see for the sphingolipids, GPI protein nanoclusters, and so on. I still believe, because we have really experimental evidence, that the lipids really are involved in these, so to say, hotspot size. Okay, so much about the lipid stuff. So we further, because I'm now in a molecular medicine environment, of course you have to deal a lot with immunology and so on. And we try, of course, to find out where is our technique useful as well. And, oh sorry, I forgot to do this. Okay, and of course, nanoscale diffusion. There's a very nice application we are working on, is HIV virus maturation. Because maturation is required for the virus uptake into cells. And it involves the rearrangement of this gag lattice in here and the end molecules. The moment it matures, the gag lattice is really pulled into the inside and the end molecules reorganize and they cluster on the surface. Now, what is not known really, how does this rearrangement of the end molecules occur? I mean, the most obvious thing is that they can become much more mobile in here because then they can move around and cluster. Now, our goal was to measure really the mobility of these envelope molecules, these end molecules, on a single HIV virus. The problem is, of course, the size of the virus particles, smaller than 200 nanometers. And you also only have 5 to 10 copies of these envelope molecules per single virus. So our approach was, we took a virus, we labeled the matrix with a GFP label, and then we had FAP antibodies labeled against the end molecules. And we now aligned them on a cover glass surface, and we nicely see these different single virus particles. And then we use scanning STAT-FCS, what I told you before. So we scanned very fast across the line, and we tried to cross several of these virus particles. And the size of our spot is 55 nanometers, so much smaller than the size of the virus particles. So along this line, this is the line, we see over time a fluctuating time trace on each of these virus particles, because molecules move in and out of this 55 nanometer large spot. We can bundle all of this data and create an average correlation data. That's what we did. We fitted with a very basic model, and what we find is, again, determining the diffusion coefficient, what we find is for the immature virus, we have a diffusion coefficient that is smaller than 0.0015 micrometers squared per second. I say smaller because a lot of photobleaching is involved here as well, so we don't really know. Maybe it's very immoral. But for the mature one, we get very nice FCS data, and we clearly can see it's roughly 0.002 micrometers squared per second, so faster. And if we take a variant of the nth molecule, so there's no interaction of these nth molecules with the GAG matrix, and also no interaction between the nth molecule, we expect a high mobility, and that's what we see. So it's even faster in these molecules. So we really have a tool now to in more detail look on the mobility of molecules on single virus particles. On the other hand, if you do research, and my unit is focused on immunology, and if you do as a microscopist immunology, you have to handle T cells. No chance not to do it. And T cells are funny cells. They have a resting state, and then when they come near an inflammation area, they're getting into an activation state. They completely polarize, and especially if they interact with an antigen-presenting cell, so a cell presenting parts of a bacteria or virus, they form a very close contact with this cell, the so-called immunological synapse. In this contact, a lot of molecules rearrange to then trigger an immune response. But of course, you don't want to trigger an immune response out of nowhere. You really have to be certain that you have a real antigen presented there. And that's why the molecular mechanisms involved in this synapse formation is very complicated, and it's a perfect tool for all of the tools that we have. Now, how was it investigated before? Usually people use turf microscopy, and they put the T cells on glass or on a bilayer, and this bilayer or glass was presenting the antigens. And then with turf, you looked over time, this is an old paper from 2005, you looked over time how these molecules rearrange, and at the end, forming the synapse. So it's a perfect tool. But to really disentangle the molecular transitions and transformations, you have to compare it to the resting state. So in turf recordings, people have done it to put the T cell just on glass. No antigens, just glass. Or inert proteins. And people have seen with super resolution also pre-clustered clustering of molecules. So we thought, let's do the same. So we've chosen the kinase LCK. LCK is one of the first molecules starting to trigger the T cell. It's really involved in phosphorylation of the T cell receptor, and thereby really starting a cascade. That's why we chose it. We took live T cells and we put them on glass. And then we looked on the glass contact. And we looked on the distribution of LCK. These are confocal, these are STAT recordings, and if we zoom in, we also see these really small clusters of LCK. But is the glass surface really the resting state? Why am I saying this? Well, there is this, we work with this together with our colleague Simon Davis on this, and he proposed this kinetic segregation model. So if the antigen-presenting cell and the T cell form a close contact, certain molecules, because they have large head groups, cannot get into this contact zone. And especially phosphatases, they have a very, like CD45, they have a large, huge head group. They are excluded from there. If you don't have any phosphatases, you disturb the equilibrium of phosphorylation. So LCK, we can really start triggering phosphorylation of the TCR. Now, if we look closer, what do we have here on the glass? We have a contact zone. So what we did is we looked on calcium signaling. So this is the signaling, this is the fluorescence of the calcium-sensitive dye, and we let these cells sink on the glass surface, and the moment the molecules touch the glass surface without antigens, they trigger. So we thought, okay, let's investigate these T cells without an interface. So we get the cells away from the surface in suspension, And we can investigate it because we don't use turf, we use confocal or stat microscopy. So we really can get away from the surface. And look what we found. We found this is the T cell, and we found a very homogeneous distribution of the LCK. Also, no calcium signaling. But the moment we added antigens, either to the solution or we let the cells sink to the glass surface, we saw calcium signaling and reorganization of the LCK. So clearly, the glass surface drastically alters the organization of the proteins in the T cells and don't allow a real investigation of the resting state. And of course, it's a further indication of the segregation model. Now, to deeper understand the molecular processes that go on the T cell activation, so far we used glass surfaces coated with antigen, but it would be great to mimic the membrane interaction between the APC and the T cell. So, going back to the work, Jay Groves and Mike Dustin, they used a lot this bilayer system. So, they used the bilayer system on the glass substrate, and they put the T cell on top. And they incorporated these antigen-presenting molecules into the membrane. But they did this usually through artificial lipid-linked protein incorporation. So those molecules don't really see a real membrane environment, these antigen-presenting molecules. But remember, we also want to investigate the influence of the lipids in here. So we couldn't do this because it's really doesn't show its transmembrane region here. So we thought, hey, let's go to our GPMV system, because the antigen-presenting molecules are fully membrane embedded in these GPMVs. And what we try now is to really create supported plasma membrane bilayers, to really drag the GPMVs onto the support so they spread on there and form a bilayer. And then in this bilayer, of course, we have all the proteins, all the lipids in there. First, we check the mobility of different molecules in there, because if there's no mobility, it doesn't make sense. We see pretty good mobility. Cholesterol, lipids, GPI-anchored molecules, even Fc receptors, transmembrane proteins. They all show pretty high mobility in there. What is left to be shown is really, do the GPMVs blast like this, or do they blast like this? So it's the outside really facing on the top, because that's what we need for our activation. Okay, I mentioned before, and this is my last point, that the actin cytoskeleton plays a very important role in all these interactions that we see, and also these hotspots and so on. So with STAT, we can also nicely investigate the actin cytoskeleton. This shows in suspension a T cell, confocal STAT recordings, and the STAT recordings you nicely see, we can really see this meshwork. This is a live T cell, but just investigating a meshwork in a resting T cell is not so interesting. What we really need to see is how does the cytoskeleton rearrange upon activation. So we did our same model. We coated the glass with antigens, and we let the T cell really land on the surface and looked at different time points of this landing, how did the actin cytoskeleton rearrange. So we fixed our cells at different time points and did STAT imaging. So this was really at the first contact. Then after a while, the actin cytoskeleton retracts to the outer part and then contracts again. And you can really see nicely follow how the actin cytoskeleton rearranges, meshwork sizes, and so on. Filament lengths we determined. There's different advanced microscopies. But with STAT, look how long we have to look the time scale, really up to 900 seconds. With a STAT microscope, it's impossible to look on a live cell event over 900 seconds. You just kill the cell. So we have the structural information here, but we don't have the dynamic information, how one state transforms into another state. So we teamed up with Eric Betzig and Janelia, and we investigated exactly these dynamics using his lattice light sheet microscope. The lattice light sheet microscope has lower spatial resolution, but the phototoxic effects are very low. So we can really look at the living cell over 900 seconds. And this is shown here. Oh, no, it doesn't show. Okay, the T cell, what is labeled here in blue and green is the actin, and this is myrcene. And as the T cell lands on the surface, you see how it spreads out, later on contracts again, and also we can see how the whole phenotype of the cell changes, height, area, contact area, and so on. And with this, combining these data with our high-resolution STAT data, we could come up with a very nice model how the actin cytoskeleton reorganizes in very much detail over this interaction with antigens on the surface. Now this shows also these examples. They show that we run also an image facility in Oxford. And we see all these different applications. Some people require high spatial resolution. I showed you that. Some require high temporal resolution. The FCS can serve that as well. We have long acquisition time sometimes we need, low phototoxicity. The lattice light sheet microscope can do this. I didn't show you 3D or deep tissue imaging, but this is also a big demand of a lot of molecules. And we have a lot of different microscopes around there that serve one or the other aspect of this. But we don't have a microscope that can do it all. That's why, for example, in our facility, we try to be very diverse with the microscopes that are in there. On the other hand, you also might want to combine it with other readouts. For example, with this T-cell interaction, what we did at the same time, we tried to investigate the forces done by the T-cell on the antigens or on the support. So you can do this traction force microscopy where you have an elastic gel full of fluorescent beads. And while the T-cell touches this is the actin, this is the T-cell, and these are the beads. And look on the lower one, especially in this area. Once the T-cell touches it, you see some bead movement. And out of this bead movement, we can determine the forces at the same time that this T-cell puts on this. So combining different readouts is also great. So what is still left to do in our microscopy measurements is really to further develop these techniques and really try to find maybe a microscope at some time that can serve really with a good trade of all of these demands. This brings me to the end. I, of course, have to acknowledge also people back in Göttingen. Especially I have to thank Alf Honigmann, who did a lot of these measurements on the lipid and lipid nanodomain stuff, and he has now his own group in Dresden. Of course, I have to thank Stefan for his continuous support, and I have to thank the lipid labeling guys. They did a tremendous job. Vladimir Belov in Göttingen, and Günther Schwarzmann in Bonn. And Günther Schwarzmann, I remember I called him up and asked him, he's a really very experienced guy in this field, and I called him up and asked him, hey, can you label us the gangliosides? Because he's a ganglioside expert. And he wanted to know what we want to do, and then he asked me, do you really want to go into the lipid nanodomain or lipid raft field? It's a wasp nest, but I'm glad we did it. So he was a very tremendous support over the years, and of course all the collaborators over the years. And now I have to, of course, thank all the people, my team. For example, Jorge did all the work on the LCK. Matthias, a lot on the building of the microscope, is just like Sylvia. Marco did all the actin work. Erdinch is really involved in all the GPMV business. Jacob is doing the virus stuff. Hugh is doing the traction force microscopy. Tess is really fighting her way in primary immune cells. So we have a great team of biologists and physics people that really can combine the technology know-how and the biological know-how. I have to especially thank Christopher Lagerholm and Dominic Waith from the image facility. They have been tremendous. And of course, our colleagues in the WIM, especially Simon Davis, with whom we do most of the T-cell work. And of course, I have to thank all the funding organization, the Wolfen Center, the MRC, the Radcliffe Department of Medicine, and the Wellcome Trust. And thank you for your attention.
Thank you, Christian, and this was excellent. And we still have time for a few questions in case. Yes, please.
A nice talk. I was wondering, could you couple the STED with the FRAP to look at lateral diffusion in the membrane?
We use FRAP, of course, as a complementary tool to look all on this diffusion, because FRAP, for example, gives you a very important information how much more immobile stuff we have in there. We're thinking about that because it would be great if you really want to photobleach only a certain part of the cell. You could be more distinct on where to look at the diffusion. We thought about this, we even started to investigate it, but of course what you would have to have is, you would have to have less photobleaching in the donut area than in the center. And let's be honest, we all know what's the problem of STAT, so that's the problem. But we're investigating different dyes, and we have one or two dyes that really, in the donut area, photobleach less.
Hello. I had the question about the supported lipid bilayers that you were trying to make from the cell blebs.
Yes.
And you showed some diffusion data about a couple of lipids and proteins.
Yes.
And were there any of what were the particular proteins that you had labeled? It kind of went by pretty quick. Were they actually transmembrane proteins, or
there
were
was
they peripheral?
one transmembrane protein, a GPI-anchored protein, because we wanted to look on all different kinds of proteins. Of course, our lipids, cholesterol, and so on, and they all seem to diffuse. But interesting, they diffuse very, very fast. Of course, you don't have an actin cytoskeleton, and the actin cytoskeleton hinders the diffusion. But they are mobile, and they are surrounded by their normal lipid environment, and that's the most important part.
Any further questions? No, not the case. Well, and thanks again, Christian. It was real excellent.